Welcome, everyone, to this week's edition of the Extracellular Vesicle Club. Today features a presentation from Felix Horns, who's a postdoctoral fellow at Caltech and in the lab of Michael Ilowitz. So this publication came out very recently, and it tackles a question that many of us in the EV field have busied ourselves with, or have at least thought about. And as we were chatting just before we admitted everyone from the waiting room, Felix mentioned to me that the MySub criteria had provoked some questions and some experimentation that was in his project, and I was really happy to hear that, because I think that one of the factors that has prevented us as a field from making more progress in understanding extracellular RNAs and their association with EVs has been this very disparate approach, these disparate approaches, I should say, to how we separate EVs and characterize them as we try to understand the extracellular RNA component. So Felix, thank you so much for being willing to share your work with us, and please go ahead. I'll leave this to you. But wait, before you start, let me just remind everyone, please put your comments and your questions in the chat box. And when Felix has finished the presentation, then I will allow unmuting, and I will call on you in order to continue the conversation. Felix.
Thank you, Ken. Well, I'm delighted to speak with all of you today, and I'm grateful for the chance to share some of our work. As Ken mentioned, I'm a postdoc in Michael Ilowitz's lab at Caltech, where we mostly work on synthetic biology. And so we've been approaching extracellular vesicles and RNA packaging from more of a synthetic and applied point of view. So we've been asking whether we can engineer proteins that induce cells to package and secrete RNAs of interest within initially virus-like particles and also vesicles. And the goal is really to address two major challenges in biomedicine. So both of those challenges are related to RNA. RNA allows us to read and write cell states, but the existing approaches for doing so have some real limitations. So we live in a golden age of RNA analysis, and revolutionary techniques like single-cell RNA-seq and in situ hybridization allow us to decipher the molecular states of individual cells. But to get to the RNA, we need to crack the cell open. And this kills the cell, and it prevents us from observing the same cell over time. And this is a major drawback, because these cell dynamics lie at the heart of fascinating and important biomedical questions, such as how embryos develop, how immune cells fight pathogens, how tumors spread, and how our bodies age. And at the same time, RNA allows us to control cell behaviors, hence the tremendous promise of RNA therapeutics. But this field is held back in large part by the major challenge of delivery. It's hard to deliver RNA to specific cell populations within tissues when and where it is needed. And so these challenges may seem completely distinct, but they can both be addressed by a single approach of RNA export. So we've been engineering living cells to package and secrete RNA within virus-like particles or vesicles. And we can then collect and sequence that RNA to see what cells are doing. And we can collect samples over time to monitor cell dynamics non-destructively. And this approach also allows one cell to deliver RNA to another cell, opening up a new strategy for delivery, where an engineered RNA delivery cell explores the body, detects target cells, and locally delivers RNA when and where it is needed to do things therapeutically like reprogramming cell states or killing diseased cells. So today I'd like to tell you first about how we've engineered VLPs, or virus-like particles, and EVs, extracellular vesicles, to create RNA export systems, and then how we've harnessed them to track cell dynamics and to deliver RNA from cell to cell. So when we started thinking about RNA export, we sat down and we realized that an ideal RNA exporter should provide several key features. It should specifically export only the cargo RNA of interest, and it would export a lot of that RNA. So it would, in other words, have both specificity and efficiency of export. And it should protect that exported RNA from degradation. and it would allow the exported RNA to enter and express in other cells when we want it to. And finally, it should minimally perturb cells that are expressing the export system itself. And we're not the first to become interested in packaging and secreting RNA. There's a rich history of ingenious systems for doing so. And this is far from an exhaustive list that I'm going to go through, but it covers some of the major classes of systems, which each have a different mechanism, and they've each had their own limitations so far. The simplest systems are natural viruses or virus-like particles. And so these systems use a non-engineered viral capsid together with the natural packaging signal of a virus. So you can take that packaging signal and attach it to an RNA of interest to more specifically load it into these particles. And so these are simple systems, but they have the drawback that many viral capsids have only modest binding specificity for the viral genomic RNA, and they readily bind other cellular RNAs, so they have limited packaging specificity. And they also inherit other limitations from viral biology, such as only packaging 2 cargo RNA molecules per particle in the case of retroviral systems. VLPs have been engineered to add an RNA binding domain to improve the packaging specificity for RNAs of interest, which are tagged with a cognate RNA aptamer. And this has been explored by a number of groups, but previous designs have often introduced mutations that are known to lead to inefficient particle assembly and secretion. And finally, EVs have been engineered by adding an RNA binding domain to a protein that is incorporated into EVs, such as CD63. But these have suffered from inefficient cargo loading and secretion, as well as restricted cargo capacity, possibly due to the small size of some of the EVs that are generated this way. And a common problem across many of these approaches is that the RNA export efficiency and specificity often have not been quantitatively characterized, and the benchmarking methods differ across studies, making it challenging to make comparisons between systems. So when we started thinking about exporting RNA from cells, we were initially inspired by RNA viruses, which package and secrete their own genomic RNA as a step in their life cycles. And intriguingly, expressing only the capsid proteins of many viruses in mammalian cells is enough to induce secretion of VLPs, virus-like particles. And this suggested that these could form the basis for a genetically compact RNA packaging system. We've also been interested in protein nanocages and vesicles. And we focused in on these designed protein nanocages, which were developed in David Baker's lab. And they're composed of designed protomers that self-assemble to form a nanoscale cage. And beautiful studies from Wesley Sundquist and Neil King showed that by decorating these cages with a membrane binding domain and a secretion signal, these cages can be secreted within extracellular vesicles. And so the membrane binding domains that we've used in this work include domains which become post-translationally modified with a lipid or a hydrophobic domain that directly inserts into the inner leaflet of the plasma membrane. And the secretion signal that we've used here is the P6 peptide from HIV, which recruits the escort machinery. And I should mention that these systems are modular in the sense that we can swap these components in and out, and we've shown that a number of these types of systems can work. And so this adding these 2 components, the membrane binding and the secretion signal, to the cage induces their secretion inside of vesicles. And this is an electron tomogram reconstruction from the Sundquist lab showing these cages, which are shown in gold, and they're about 25 nanometers in diameter, packaged inside of an enveloped vesicle. So this is a large, 120 nanometer vesicle. And so to package RNA into VLPs or vesicles, we fused some of these different proteins to RNA binding domains. And we've explored a variety of these RNA binding domains, and I'll focus here on the classic MS2 coat protein, or MCP, which is from the MS2 RNA phage, and it's actually the capsid protein of the RNA phage, which it uses to package and secrete its own RNA from bacterial cells. And so it binds with high affinity and specificity to an RNA stem loop aptamer called the MS2 stem loop. And we tagged cargo RNA with an array of these MS2 stem loops. So we've actually created dozens of designs based on either capsids or nanocages, some of which are shown here. We started out with these natural retroviral capsids, known as GAG proteins from a relative of HIV called MMLV, and then later we moved to engineering the HIV capsid. We then engineered them to have the MS2 coat protein as a fusion, and we further engineered them to remove their natural RNA binding domains, producing these GAG-ZIP variants. And with the nanocages, we've tested a number of different architectures which have different membrane binding domains and permutations of the domain ordering. So we started with the GAG capsid protein from MMLV and tagged a cargo RNA with its packaging signal called Psi. And we transfected these two components into HEC293 cells, and we sequenced the total RNA secreted into the supernatant, either in conditions including or excluding the exporter, which in this case is the GAG protein. And we found that this system exports the cargo RNA efficiently, but not specifically. So the tagged cargo RNA is enriched more than 1,000-fold in the presence of the exporter compared to its absence. That's indicated by the pink star. However, essentially the entire transcriptome was similarly enriched, as shown by the heat map being off diagonal. And this can be explained by the non-specific RNA binding activity of the GAG nucleocapsid domain, which was previously shown. And so iterative engineering of the exporters, especially their RNA binding interactions, enabled efficient and specific export. So we added the MS2 coat protein, forming GAG-MCP, and then we removed non-specific RNA binding domains from the capsid, forming GAG-ZIP-MCP. And finally, we replaced the capsid entirely with the nanocage, which lacks a natural RNA binding domain. And these progressively more engineered exporters maintained efficient export of the tagged cargo, while their specificity was dramatically improved, with non-specific export reduced to nearly undetectable levels. And so just to highlight the specificity and efficiency of these exporters, with the most efficient and specific variants, most of the secreted RNA was the cargo. So with this nanocage design called EPN24 MCP, 81% of the RNA in the supernatant was composed of the cargo RNA. So we quantified the absolute rate of export by measuring the accumulation of RNA in the culture supernatant by RT-qPCR. And we found that these exporters secreted about 1,000 molecules of cargo RNA per cell per hour. And this is about 1,000 times higher than natural rates of RNA accumulation due to natural secretion processes or cell death in a HEC293 culture. And so cells expressing these exporters secreted either virus-like particles or extracellular vesicles. And I wanted to highlight that these vesicles, so focusing now on the nanocage-based vesicles, they were secreted via the escort pathway as designed. So when we co-express the system with a dominant negative inhibitor of escort-dependent secretion, export was entirely suppressed, showing that escort activity is necessary for secretion. And conversely, factors that enhance escort-dependent vesicle release also enhanced RNA secretion. So it was previously shown that HIV release is enhanced by overexpression of factors that allow escort to be recruited more efficiently to sites of viral budding. And because our vesicles use this P6 domain from HIV to recruit escort, we hypothesized that release of the vesicles could be similarly enhanced. And so we co-expressed the system with a panel of modulators of viral or exosome release. And two of these modulators, which are known to enhance escort-dependent HIV release, increased RNA secretion by up to 4-fold. And by contrast, the other modulators, which are not known to interact with escort, did not affect RNA secretion. And so we call these factors boosters, and they enhance the release of vesicles and thus RNA. And so these exporter particles protected RNA from degradation. When we challenged supernatant containing exported RNA with RNases, the RNA was completely protected from degradation, and that's shown by the red arrows. By contrast, when detergent was added, this protection was abolished, showing that the RNA is really packaged inside of lipid-enveloped particles. And the RNA was also protected during incubation in whole blood at physiological temperatures. So we took RNA packaged inside either VLPs or vesicles, spiked it into whole mouse blood, and incubated it for 24 hours. And we found that there was no detectable loss of the exported RNA, in contrast to free unpackaged mRNA, which was rapidly degraded. And finally, an ideal RNA export system should minimally perturb the cells that express it. And so we initially monitored perturbations by just looking at cell morphology. We found that there were no detectable differences between cells expressing exporters and control cells expressing only fluorescent proteins. And this is not shown here, but we've also found no differences in the growth rate or the abundance of dead or dying cells. And to look for more subtle perturbations, we sequenced the transcriptomes of cells expressing the exporters. And here, again, we found no perturbations associated with exporter expression. So we're looking at differential expression analysis, comparing cells with and without the exporter. And in every case, we found no differentially expressed genes that were consistent across replicates. And so this indicates that the exporter expression does not detectably perturb the cellular transcriptome. So to sum up this section, we engineered exporters to package, secrete, and protect RNA from living mammalian cells. And from an engineering point of view, we'd like to propose that this quantitative framework for measuring RNA export efficiency and specificity using qPCR and sequencing will be useful for benchmarking and comparing between current and future systems. And we think these dual metrics for efficiency and specificity, namely efficiency measured in units of RNA exported per cell per unit time, is really the fundamental unit to use. And specificity is ideally measured using sequencing. And so next, we asked whether we could sequence this exported RNA to obtain information about cell populations in a non-destructive way. And as an initial demonstration, we tracked cell population dynamics by exporting and sequencing clonal RNA barcodes. So the concept is to label each cell within a starting population with a unique barcode, which is inherited by all of the descendant cells that form a clone. And as the clones grow, they transcribe and export their barcode. And as a result, the abundance of each barcode in the culture supernatant reflects the population size of the corresponding clone. And sequencing these exported barcodes allows monitoring of growth and death of the cell populations. And so to implement this, we engineered a human cell line to inducibly express an RNA exporter. And we prepared a diverse lentiviral barcode library, which delivers an exportable reporter transcript that contains the barcode. So when we induce the cells using a small molecule, they will export these clone barcodes. As a demonstration, we tracked population dynamics during drug selection. We prepared two different drug-resistant cell populations so that drug selection could provide a ground truth of which populations should expand or contract. And we barcoded these populations with two different lentivirus libraries, which have different sets of barcode sequences. We sorted 5,000 cells of each population together into a single well. And we cultured them in the presence of one drug or the other while collecting supernatant at daily time points and sequencing the exported barcodes. And so these barcodes revealed population dynamics in the culture. So taken as a whole, each drug-resistant population grew exponentially, while the sensitive populations contracted. And within each population, we resolved thousands of clones. And tracking these individual clone barcodes revealed the clonal population dynamics of drug selection. So this plot shows the relative abundance of 100 clones randomly sampled from the several thousand detected within each population. And we can appreciate that individual clones within the drug-resistant populations expanded exponentially, whereas the sensitive clones contracted. And we could even see clonal extinction events where barcodes disappeared after a certain time point. And so these results demonstrate that RNA export enables non-destructive monitoring of cell population dynamics with clonal resolution. And we're starting to extend this from a culture context into animal models for tracking clone dynamics in contexts like cancer and development by sampling RNA barcodes from the blood. And so in the last few minutes here, I'd like to briefly explain how we've also used these exporters to deliver RNA from cell to cell. So for delivery, we return to viruses for inspiration. Viruses enter cells using proteins called fusogens, which fuse the virus and cell membranes. And so we wondered if we could endow these exporter particles with the ability to enter cells and deliver RNA by expressing a viral fusogen on their surface. And we started using the classic fusogen VSVG, which is often used to pseudotype lentiviruses. And we added it to the nanocage-based export system. And this was work done together with a fantastic grad student, Joe Martinez. So for testing, we used a Cre-based system. So we delivered an mRNA cargo encoding the Cre recombinase. And as receivers, we used Cre reporter cells, which activate a fluorescent protein upon Cre recombination, which provides a sensitive way to detect functional activity of the Cre mRNA. And with all of the components present, a majority of the receiver cells had Cre recombination, while negligible delivery was detected when we left any one component out. And this indicates that decorating these exported vesicles with a fusogen enables efficient delivery and expression of RNA cargo. And as an aside, we did similar experiments using the VLP-based systems, for instance, GAG-MCP, and we found that there was very little expression of the cargo RNA upon delivery. And we think this may be because the nanocage structure is an open and porous structure, which allows access by ribosomes to translate the mRNA after delivery, right after cell fusion, whereas with the virus-based designs, there needs to be disassembly of the capsid before the RNA can be translated, which is an inefficient process. And the other thing I wanted to highlight is that in this result, the supernatant was not concentrated. So this is just transferring crude supernatant from one plate to another. And so this really shows that this is a quite efficient platform for delivery of RNA. And the ability to deliver 2 RNA cargos simultaneously would allow us to transfer more complex RNA circuits. And so to test this, we tried delivering 2 fluorescent protein encoding mRNAs. And so we found that both of the colors here were successfully delivered to a majority of cells. And in these experiments, we're making use of these export enhancers, which I described previously as enhancing the export of RNA. And we found that they also enhance the delivery of RNA in proportion to their export. And so finally, to test whether the system can deliver RNA directly from one cell to another, we tested delivery in a co-culture setting. So we've established a system where sender cells are localized in the periphery of a well, and the receiver cells are in the middle. And the sender cells in this setting successfully delivered Cre RNA to receiver cells throughout the population, really demonstrating direct delivery of mRNA from cell to cell in a co-culture. And this is quantified at the top there. And so we named these systems Courier for controlled output and uptake of RNA for interrogation, expression, and regulation. And to summarize, we've developed these genetically encoded RNA exporters inspired by viruses and extracellular vesicles, which package and secrete RNA from mammalian cells. And we've shown that sequencing exported barcodes enabled non-destructive monitoring of cell population dynamics, and that these exporters can deliver RNA from cell to cell. And so we hope that these RNA exporters will form a foundation for new types of reporter and delivery systems to help address the major challenges of measuring and manipulating cells. And so I just want to close by thanking my amazing mentor, Michael Elowitz, our RNA export team, and thank you for your attention. And I'd be happy to continue with more discussion.
Felix, that was a wonderful presentation, very, very clear. And, you managed to condense an amazing amount of work into, into a bite-sized presentation here that we can digest. So thanks for that. And I am going to go ahead now and allow unmuting, and also for people to share their videos if they like. So if you have not put your comment or your question in the chat box, could you please do so? And I will get to you in the order in which your comments are received. So I just wanted to start out with a question for you, Felix, about the RNase protection assays that you did. I may very well have missed this, but did you do those assays with a prior protease treatment, or were they with RNase alone?
That was with RNase alone.
Okay.
Yeah.
That's something that we found on occasion, and I mean, we would assume that these RNAs would be internal, but in some cases they can be protected by proteins on the surface. We don't even know the mechanisms whereby these RNAs would get out of cells. Maybe they're just from dying cells. But sometimes that a prior protease treatment will expose certain extracellular RNAs that are there.
It's something that we've thought about quite a bit, because we observed something interesting with the VLP based systems. So we actually think that there are two levels of protection for the RNA. Both the envelope protects it, and protein binding the RNA itself protects it. And the reason we think this is because with that RNase protection assay, when we add the detergent to the VLP-based systems, we found that some of them have some residual protection even after the envelope has been removed by detergent. And we found that interestingly correlates with the degree of non-specific RNA binding of the protein component, meaning that these capsids which non-specifically bind to RNA, we think they basically tile along the cargo RNA and provide protein level protection to the RNA even after the particle is removed, or even after the envelope is removed. So that's something we've thought about following up on to really understand whether there is this second level of protection, and adding a protease would be an ideal way to test that. So that's something that's on the to-do list.
Very good, thank you. All right, we have a question here from Zach Troyer. Zach, please go ahead.
Can you guys hear me okay?
Yep, sounds good.
Great talk, Felix. It was really interesting. My question just relates to the viral fusogens. I was just curious if you've thought about using alternative fusogens to VSVG, given that it's relatively cytotoxic.
Yeah, so we've thought a lot about that, and we've started exploring other fusogens, which I just didn't show. I think there certainly is an issue with VSVG coming from this cytotoxicity. I think, our goal of creating this cell-based RNA delivery requires having a fusogen which is not cytotoxic, because we can't kill the sender cell in the process. And the other issue that can come up is VSVG causes cell-cell fusion. And so we wouldn't want the sender cells to be fusing with receiver cells directly. And so we've certainly been interested in exploring other fusogens, including ones which are less cytotoxic or ideally wouldn't cause cell-cell fusion. And we've tried some other viral fusogens, and we've shown that the system is modular in the sense that we can use other fusogens with it. And we've been playing around with different natural fusogens, as well as recently there have been a number of engineered fusogen systems. You may have seen these, where you can direct the specificity of fusion using some sort of attachment domain or affinity domain, like an antibody, to kind of redirect particle entry in more of a programmable way. And so we've also started playing around with those, and those also work with the system. So we think that there's kind of a large space to explore different fusogens, which might have less cytotoxicity, as you suggest.
Thank you.
We have a couple of questions here from Karthik, whose microphone is not working, so I'm going to go ahead and ask these. The first question is: Have you made some cell lines to express, your constructs?
Mm-hmm. Yeah, we have. So we've made a variety of different cell lines, and I should mention that the RNA, the population dynamics reporting experiments, which I showed, were done with stable cell lines. So those lines had an exporter integrated, which is on a DOX-inducible promoter. And we've also integrated the cargo into those. So these systems do work in stable cell lines.
Great. And so if Karthik or anybody else who's watching this wants to get a hold of those lines, is that possible?
Yes, certainly. Feel free to email me, contact me offline, and we can make that happen, certainly.
Awesome. So the second question that Karthik has is about the RNA and how it's going to be functioning in the recipient cell. So how is RNA released into the recipient cells from this MCP construct that you reported?
Yeah. So it's an interesting question. So the question is, after RNA is delivered from the sender cell to the receiver cell inside of this capsid formed by GAG-MCP, how is the RNA released in order to become functional? So that's actually, I think release happens quite inefficiently in the case of GAG-MCP. And we think that because when we try to do these delivery experiments using the capsid-based systems, we see very little expression of the cargo. And we think what's going on is those capsid-based VLPs are able to fuse with receiver cells, but the RNA does need to be released, or the capsid needs to disassemble in order for the RNA to be translated. And we think that process is inefficient, which is what causes poor expression. And the contrast is with these nanocage-based delivery systems, where after cell fusion, those cages should be released into the cytoplasm. And because those cages are an open structure, they're actually quite porous. We think that the RNA is available for translation without requiring disassembly of the nanocage. And so we think that contributes to the higher efficiency of expression of the cargo that we see with the nanocage-based systems.
Interesting. Yeah. So you think that it's actually with the GAG-MCP, the rate limiting step there is the disassembly of the capsid structure.
That's what we think.
And not release of the RNA per se.
Yes, we think that's the case based on the literature on viral entry and initiation of viral gene expression, because that is the case for many RNA viruses, where what's required is disassembly of the capsid prior to expression. And in the case of these GAG-MCP systems, this is actually what's called an immature HIV particle, which is known to disassemble inefficiently.
Great, thanks for that answer. Elena Martens, you have several questions here. Please go ahead and share those with us.
Yes, thank you, Ken. And Felix, also thank you for a great presentation. I hope you can hear me, guys. I do have two questions, but your last comment intrigued me quite a lot. You mentioned that you think that the translation of the RNA happens inside the nanocage. You think? Did I understand it correctly?
That's what we believe, but this is something that we're working on getting more direct evidence for. because what we'd really like to see is a co-localization of the cage and the RNA as it's being translated. And
the ribosome.
yeah, exactly, and the ribosome, exactly. And so we don't have direct measurements of that, but that's our model.
Okay, that's very cool. If you can show that, that'll be really great to see. Okay, actually, I had a slightly different question. Actually, I was wondering, do you have any size limitations on the transcripts, and do you need to modify GC content or any other little tricks in that sense?
Yeah, so the first question is there a packaging capacity? We have tried to measure the packaging capacity, and we've found we've been able to export transcripts up to 10 kb in length, which are the longest ones that we've tried. And we're working on testing longer transcripts to really define the upper limit of the packaging capacity. Your second question is about a related question about, is there anything about the transcripts that allows them to be packaged more efficiently?
Yes.
We haven't tried playing around with that. It's certainly something that seems like it could be tuned or optimized. Things like GC content are not things that we've tried to tune, but that's certainly something that I think is worth exploring. I will say that my expectation going into testing that type of thing would be that the packaging is mainly driven by this MS2 stem loop. And so I'd expect that the dominant effect is just whether or not you have those loops and how many of them you have. Because we've shown that if you have more of them up to a certain point, you get higher export until it saturates with a certain number of repeats, 8 repeats. So I would expect that, you know, most of the packaging efficiency will be driven by that signal rather than the sequence of the rest of the transcript. But that's something that we want to try to define better.
Okay, thank you very much. Great talk and nice discussion also. Thank you.
Thank you.
Thank you. I want to take us back to the kind of the biogenesis side, the very beginning of all of this. You mentioned the amount of RNA that is released per cell. Can you then make a calculation as to the percentage of released particles that contain the RNA, the percentage of released particles that, in the case of the fusion proteins, that would have a fusion proteins on the surface, and then if, you know, how many of those particles are actually double positive? You know, they've got an RNA on the inside and they have, you know, some sort of surface moiety.
Yeah, that's a great set of questions. I think, you know, defining on a per particle basis what the loading efficiency is and also the loading efficiency for the fusion, these are both really important questions for kind of characterizing this, especially as a drug. We are working on those types of measurements. I think the simplest thing is to measure how much RNA there is in the supernatant and measure how many particles there are in the supernatant. And this lets us get not exactly what you're asking for, which is on a per particle basis, you know, how many are full and how many are empty, but it lets us get an average number of RNA molecules per particle. And so we've done that type of bulk measurement, and we've estimated that there are about 3 mRNA molecules per particle, but that's a measurement with pretty big error bars. So we're working on refining that. As far as on a per particle basis, you know, how many are full, how many are empty, that's something that we're really interested in. And it would require, I think, going and looking at individual particles, which is something that we've tried to do a little bit by just laying them out on a slide and using in situ hybridization to probe the RNA inside of individual particles. And so that's something that we'll hopefully have an answer for soon. And that's also looking at the fusogen on those same particles, I think that's a fantastic question. And it's also something that we should also be able to do using an antibody against the fusogen in a similar imaging setup.
Yeah, very, very interesting. Yeah, so we've been doing some work with viruses and virus-like particles in my lab. In fact, Zach Troyer, who's on the call, and I also see Mona Batish from University of Delaware here, have been doing some single particle imaging with RNA. So it's oh, hi Mona. I see you just do you want to make a comment perhaps?
Yeah, no, I was about to ask that question to you, like, because if you could see them, because the good thing about SM fish is you can see even if there are 1 or 2 particle, or if the thing is empty. So that would be a good idea to really get a sense of what is the copy number being packed in each one.
Yeah, that's fantastic. And I'd love to touch base about strategies for really doing that single particle imaging in a way that's as sensitive as possible, because that's one of the limitations that we seem to encounter, is that when we do the in situ assay, we're not confident that we're detecting all the RNA which is present, because when we do a separate measurement by qPCR, we get an answer which is more RNA than what we see by fish. So, we're trying to figure out how to really get a sensitive assay.
So when we started SM fish a while ago, we are about 85% sensitive, like we're as sensitive as a qPCR, and we could pick about 85. Only thing that you would miss is something that is being translated actively at the site, and that region is being occupied by ribosomes. But the probes are, I don't know if you use RNA probe or you use SM fish.
Yeah, we've tried a variety of including traditional single molecule fish, as well as hybridization chain reaction. So
I see.
Yeah.
No, definitely we can touch base, and I can totally help you troubleshoot, because this is something we do every day. And we pick, even if there is like 1 copy, we see it, so it's very sensitive, and we can be a little less than digital PCR, maybe, we are now comparing them with digital PCR counts, but we pick about 85% of the total cellular RNA that's in there.
Yeah, that's fantastic. Yeah, I'd love to, I'll check it with you after.
Sure, yeah.
Yeah, so those long RNA molecules are particularly well suited, I think, for the detection. Yeah, so if you're getting good incorporation of even up to 10 kilobases, that's plenty of real there to set down your probes. Yeah. I think it's also quite interesting, that you have the capsid structure, but then you also have the nanocage, which you're essentially reporting as much looser, and whether there might be any detection differences between those 2 packaging methods. That would also be interesting to explore.
Yeah, we're definitely interested in that. And I think my comment on that would be, with these virus-like particles, one of the interesting things is that I mentioned these nucleocapsid domains of the capsid often have non-specific RNA binding activity. And so when we first started playing around with these in situ hybridization experiments, we ran into issues because the probes were binding non-specifically to the capsids. And so that's one area where the experiments are cleaner with the cages in our hands so far. But that's also something that it would be great to see if we could learn some tips on debugging, that kind of thing. Blocking the non-specific RNA binding activity is something that we've thought about, but we haven't really ironed out a good strategy for.
Very interesting. All right, so we do have another question here from Dan. Dan, do you want to ask your question?
Yeah. Hey, Felix. This is Dan over in Neil King's lab. I think we met maybe via email once. Super excited to see you're working on this, by the way. So one of the complicating things in my PhD, which kind of set me back at least 18 months, is that when we went to look for EPN7 via cryo, we found a lack of cages. When you're marketing something as enveloped protein nanocages, that can be a bit disconcerting, even though for the general EV field, it's still EVs that export things, right? Do you think that these EPN24 MCPs are still in cages, or are they maybe in trimers, which is what we saw with EPN7? It was trimers decorating the inside of the vesicle. What do you think? Have you done any cryo?
Interesting. Yeah, that's it's fascinating to hear about some of those, more careful structural characterization of what's going on in there. I think we don't know whether the cages are intact inside these vesicles. We have done some negative stain electron microscopy, and there we don't have the resolution to see whether the cages are intact. And so I think the next thing to do is, as you suggest, doing either cryo-EM or tomography to try to reconstruct what's inside these vesicles to see if the cages are really there. So, we don't have any direct evidence that the cages are intact, but, it'd be great to touch base on how exactly we could define that. We haven't tested EPN7, and, we tried EPN 1, 11, and 24, and, 11 and 24 worked well for exporting RNA, but 1 did not. It was noticeably less efficient than the other two. And so I think there may also be a dependence on exactly the architecture of the cage construct, in terms of what's secreted and how much is secreted.
That's exactly what I've seen too.
Mm-hmm.
Our current theory is that the stronger membrane binding motifs present on everything but EPN1 might be biasing them away from the cage structure, right? The cage formation strength towards like membrane, like satisfying the membrane binding motifs, but that's an N of 1 hypothesis. So
cool. Yeah. So there might be a competition there. That's
yeah,
cool.
Which could complicate any sort of hypothesis based upon like cage formations thing and like cage dynamics for the RNA binding and protection is kind of what I'm getting at it. Yeah.
Yeah. And then I think another aspect which could be complicating is when we use these MS2 coat proteins as the RNA binding domain, those are capsid proteins from an RNA phage. And so those also self-assemble to form capsids. And so there's also the possibility that oligomerization process is driving assembly of some type of particle which is not a cage, or perhaps some sort of hybrid between a cage or a capsid. So there are a lot of competing forces going on here.
Cool. Great presentation. Thanks.
Thank you.
Thanks for the question, Stan. Now I think we are at the end of our questions in the chat box. I still have a couple though. One of those is we've talked about how large of a piece of RNA you can put into your particles. What about on the smaller side? It seems like the EV field anyway has focused not exclusively, but almost so on microRNAs. Does your work have any implications for microRNA delivery, or are they just too small or too, hard to detach from whatever you would be using for the packaging?
Mm-hmm. Yeah. So the question is, could we package microRNAs, and what are some of the limitations there, or the opportunities or limitations there? We've thought a lot about packaging small RNAs, siRNAs, microRNAs. We have not had any issues packaging much smaller RNAs than the mRNAs that I showed. So we've shown that you can take non-coding RNAs transcribed off a U6 promoter, and those are packaged just fine. So this goes down to, I think the smallest we have is about maybe 150 bases. So I think there's no limitation in principle on packaging small RNAs. However, there is a limitation on specifically with microRNAs having both the packaging signal, the MS2 stem loops in this case, together with the rest of the microRNA in a way that allows us to deliver it from one cell to another, package it and then deliver it from one cell to another. And I think the limitation really comes from, specifically with microRNAs, they're going to be processed in the sender cell. And so that would, by default, it would chop off some sort of tag that you put on there to package it. In the most naive way of thinking about it. And so that would make it a challenge to package microRNAs. So I think we've thought a lot about delivery of microRNAs, because this is, of course, an appealing class of therapeutic. And so the challenge there is to design a system where we can have the microRNA remain unprocessed and keep packaging signals on it until it's packaged, and then be processed in the receiver cell so that it becomes active. So that's something we think about, and I'd love to engage about that.
Sounds great. Well, Felix, thank you so much for sharing your work with us, and we want to wish you the very best as you continue these things and as the technologies develop more. Thanks to everybody who joined today and for all the good questions and discussion. So Hope everybody has a great rest of the week, and we'll look forward to seeing you again on a future EV Club. Thanks, everyone.
Thanks, everybody.